In this presentation, Erin and Emily will demonstrate how to first make an extract containing catechol oxidase from potato, and then how to measure oxidation of catechol to benzoquinone using a spectrophotometer. So this first video demonstrates how to make an extract from potato. So you can see Emily first weighs out the 20 grams of potato, around 20 grams of potato that are necessary for this extraction. She then calculates the appropriate amount of phosphate buffer that would be required for making this extract, assuming that we need 2.5 milliliters of phosphate per gram. So she measures the required volume of phosphate buffer using a graduated cylinder. And then combines that together with the potato. Note that for this step, you want to use cold phosphate buffer to prevent any reactions from it. There tends to be a stubborn piece of potato in this particular extract. Ignore that for the moment, but essentially you don't want to overdo it because you'll end up with mushed up potatoes. So we're going to call this done. So the next step would be to strain your extract through a cheesecloth, which you can see Emily doing now. Once that's done, she will press out the extract from the cheesecloth into the flask, and then put the extract on ice to keep it cold to use the rest of the extract. At this point, the extraction is complete. It is ready. All right, the next part would be to actually set up the enzyme reactions themselves. So in your manual, you'll see there are 3 tubes, BW, PC, and CW, which Erin has labeled on the paper towel here next to the cuvettes. So as I start playing the video, you can see these are the reactions we're setting up. He will start by pipetting 2 milliliters of phosphate buffer. Because it's 2 milliliters, you can't really use a micropipette to pipette the entire volume, so you have to split it into 2 portions of a milliliter each. So he's setting the micropipette, if you want 1,000 micropipette, he's setting it at 1,000 microliters or 1 milliliter. Placing the tip, remember, just like in the first lap, in order to pipette the volume up, you first only go to the first stop on the micropipette. That ensures that you collect the required volume, and then when you release that volume, you go to the first step and then to the second stop to the left. You will do that twice for every cuvette. It is always a good idea to pipette the same thing to all the cuvettes first, because then you can use the same tip. Saving on tips, but do make sure to release to the second stop so that you don't have any left over. Alright, now he's done with the phosphate. For this, we use room temperature extraction. Right, so the next thing he will pipette the water for one of the cuvettes, The volume here is different, so it's 0.28 milliliters, 280 microliters. Same thing, he'll place the tip on, following into the first stop, the water up, and then release it into the PW tube. Okay, next is catechol. So it's again 0.2 milliliters, so his setting is already correct. What he needs to do is just put the tip and then go and get the catechol tubes PC. The next step is to add the extract. Got a bit of a technical difficulty. It's split it into two videos. But there's 0.22 milliliters of extract. Add it to tubes PW and PC. So 220 microliters, 0.22 milliliters. And add it. Catechol, not the extract. Put that on time. The extract is the thing you prepared from the potatoes. So it's now 0.22 milliliters into PW. Point 0.22 milliliters into PC. And then tube CW will get 0.22 milliliters. So as you're setting up these tubes, you should be thinking about what do you expect will happen in these tubes, depending on whether they have the enzyme, whether they have the substrate. So there's different combinations of substrate and enzyme in the different tubes, and depending on whether they have everything they need, the enzyme reaction can proceed. Think about that as you tubes. So Aaron is now mixing all of the cuvette to make sure that the solution is uniform. And then after this, you can start your 10-minute timer for your visual observation. Right, so this is what the tubes look like at the beginning of your assay. So in this slide, we're just showing you two images of what the cuvettes that Aaron just set up in the previous slide look like, either at the beginning, so at time 0 minutes, or at the end of the 10 minutes. So based on the tubes or in the cuvettes that you see in these images, make your visual observations for what you think make your visual observations for what occurred or what you can see, and then think about why it occurred. So do they have catechol? Do they have the enzyme? So think about that. Do you think any of the tubes that didn't develop color would develop color over time? So those are the questions you should be asking. We will now use the tubes that we generated in the previous slide to calibrate the SpectraVis and set up the lambda max value that we will need for such measurement. So the tube that's used to calibrate is CW. That's the tube that contains no extract. That's sort of zeroing out any background contribution from the catechol that's in the solution. So calibration takes a couple of seconds. Once that's complete, we will use tube PC, which contain both extract and catechol and developed a nice brown color, to obtain a full spectrum and then find our lambda max, or the wavelength at which the absorbance is high. Calibration is now finished. So put the tube CW away, and then pick up tube PC, wipe it down, make sure to place it with the clear side of the light path, and then collect absorbance to collect the full spectrum of the tube. So you can see the peak absorbance happens to be around 400 nanometers. So Erin will go through the steps to actually use that wavelength for our subsequent measurement. So this is where you pick your lambda max. So now all of the readings, as you can see in the bottom right corner, will be collected at the wavelength that Erin has selected. So now it's time to actually acquire our measurements for the reactions. So we didn't show the steps to actually set up the 3 cuvettes. So that's just a matter of pipetting, similarly to the way we did for the 3 tubes in a couple of slides ago. But you can see the 3 tubes are set up and they're already starting to develop color. So it's important to begin measurements as soon as you finish setting up the cuvettes. It's very important to make sure cuvettes are mixed, which is what Erin is doing first, so that the solution is uniform. And then the other important thing to remember here is that we want to read all 3 cuvettes one after the other. So you don't want to measure just the first cuvette for 10 minutes and then go on to the second, because by that time your second cuvette will not be in the same state anymore, right? The reaction would have for 10 minutes already. So we record the value read out in the bottom right corner here for the first cuvette, then for the second cuvette, and then for the third. Sometimes it takes a couple of seconds for the absorbance to stabilize, so make sure you don't record immediately after putting in your cuvette in. So once you have your 3 values written down, then you wait 1 minute and then you repeat. So you record all 3 cuvettes at every minute. You proceed to do that for 10 minutes or until your values kind of become constant and you've reached the plateau value. So this is this way you will acquire measurements for tubes A1 through A3, and then you would repeat a similar procedure, but with the tubes B1 through B3, which will have an unknown added to those tubes. So if you're doing this lab remotely, a table of values will be provided for you so you can continue with calculations for the rest of the lab. So thank you once again to Emily and Erin for helping record these videos. And this is it for Lab 2.